Good afternoon, and thank you for joining the webinar. I'm Matt Roth with Baylor College of Medicine, and I welcome you to today's presentation. The Extracellular RNA Communication Consortium is an NIH Common Fund program which works to advance the science and research of extracellular RNA. The consortium hosts monthly presentations on a variety of research topics, and I'm pleased to introduce today's speaker, Nicole Noren Hooten, from the Laboratory of Epidemiology and Population Science, National Institutes of Aging and Intramural Research Program at NIH. The title of her presentation is Aging Affects Extracellular RNA and Extracellular Vesicles. Nikki?
Well, first off, thank you for giving me the opportunity to talk about some ongoing research projects in our lab. So as you mentioned, I work at the National Institute on Aging. So we're interested in aging because it is a major risk factor for most chronic diseases, including cancer, neurodegenerative diseases, and heart disease. How do we tackle the complex and complicated problem of aging? So I am part of the basic science unit within HANDLES, which is the Healthy Aging in Neighborhoods of Diversity Across the Lifespan study. And we take what we call a bench to neighborhood approach, whereas things that we observe or observations in the clinic in the neighborhood, we test the mechanisms at the bench and vice versa. HANDLES is an interdisciplinary longitudinal epidemiological study of health disparities in aging in Baltimore City, and it examines the effects of race and socioeconomic status on health outcomes in a cohort of urban adults. There's over 3,700 community dwellings, both African American and white, that were aged at baseline between 30 and 64 years. And we're currently getting ready to start wave 5 of data collection, and the study's been going on for approximately 13 years. And what's unusual about this study is that mobile medical research vans, as pictured here, go out into the local Baltimore neighborhood. And this enables the platform to recruit and retain non-traditional research participants into age-related clinical research. And HANDLES tackles the problem of looking at aging at multiple levels, including the social level up here, and also the HANDLES bench unit focuses on looking at aging as far as the organ, cellular, molecular, and genomic level. And so today I'm going to tell you 4 different stories about how we're looking at aging, and that I'm going to focus initially on how non-coding RNA is changed in aging from peripheral blood mononuclear cells, and this will focus on microRNAs, although we also have projects in the lab looking at long non-coding RNAs, but I'm not going to tell you about those today. And we also have projects that have looked at serum microRNAs and also extracellular RNA in aging. And lastly, I'll tell you about plasma extracellular vesicles and how they may change with age. We became interested in microRNAs in aging a number of years ago because of data first in C. elegans, where there were changes in microRNA expression that occur over the lifespan of C. elegans. And that if you change a single microRNA in C. elegans, it can affect the worm's lifespan and health span. And also, it was observed that there's decreased microRNA expression during cellular senescence in vitro. Cellular senescence recapitulates many aspects of cellular aging and is often used as a model of cellular aging. So here's just an example here of a picture of proliferating cells and senescent cells. So as many of you know, but I'll just remind you, microRNAs are short, non-coding RNAs, and they're key regulators of gene expression, where they regulate mRNA stability and translation by fine-tuning gene expression. So we initially looked at microRNAs with aging by obtaining peripheral blood mononuclear cells from young and old individuals that were in the HANDLES cohort. And the young were 30, and the old were 64. And here's a heat map of the data that we obtained looking at real-time PCR, by real-time PCR, where red indicates it's microRNAs were upregulated in old individuals, and blue is downregulated in old individuals. And what we found was that the majority of microRNAs are down in, or decreased with human age. So here's just a cartoon of this, where if we look at microRNAs, they decrease in abundance with human age. So we wanted to further define the mechanism for decreased microRNA levels with human aging. So one of the major processors of microRNAs in cells is dicer, is the protein dicer. And what we found is that dicer levels actually decrease with human age. And at the same time that we were doing these studies, here at the NIA IRP, Rafa de Cabo's lab was also looking at a diabetes drug called metformin. And what's interesting about metformin is that it increases the health span of life. And so we became, and there's many health disparities in health span and lifespan. And also a number of handled participants are taking metformin. And what we found was that metformin actually increases DICER levels. And this causes an increase in the subset of microRNAs. And that metformin is currently actually being proposed and is in clinical trials as an anti-aging agent. So what we think is that metformin in part increases DICER levels. This increases a subset of microRNAs, and this causes a resistance to stress, aging, and cancer. So we first have looked at microRNAs in relation in their profile in peripheral blood mononuclear cells. And we next wanted to ask, do microRNAs change in body fluids? Now, I don't have to go into too much detail about to this audience as far as where extracellular RNAs are found, and that they're found in majority of body fluids that's been tested. But here we're going to focus in our lab on plasma and serum, as plasma and serum are very readily available. So circulating microRNAs are highly stable and they're resistant to harsh conditions, and they're secreted or shed from cells, extracellular vesicles, or bound to RNA binding proteins. And microRNAs in serum and plasma have enormous promise to be used for diagnostics, response to treatment, disease suppression, and a variety of different age-related diseases, including cancer, cardiovascular disease, and neurodegenerative diseases. And as I mentioned, serum and plasma are readily bioavailable. And in the HANDLE study, we have banked samples from the 5 different waves of study of clinical samples, and also longitudinal samples from the different time periods. So we asked the question, can we identify microRNAs in serum from young and old individuals? And also, does serum microRNA expression change with human age? So to answer these questions, we obtained serum from 11 young 30-year-olds and 11 older 64-year-olds that were handled participants. We isolated RNA from the serum and performed Illumina small RNA sequencing. And then we followed this up by also doing real-time RT-PCR validation in a larger cohort of n equals 20 per group. So at this time, a number of years ago, we detected 653 microRNAs in the serum from young and old individuals. And we found that there are 143 that were downregulated with age and 106 that were upregulated. And I'm also going to focus on the three microRNAs that we've validated by real-time PCR and also pursued more in the study. So these microRNAs were miR-151A3P, 1248, 181A5P. And all 3 of these microRNAs decreased in the serum of older individuals compared to young individuals. And this was done by looking at by doing real-time RT-PCR validation in the larger cohort. And what's unique about being here at the NIAID was that we also were able to obtain serum from young and old Rhesus monkeys. And we wanted to validate whether the results that we were finding in humans can also be observed in non-human primates. And what's interesting about Rhesus monkeys is that they age 3 times that of humans. And so we can use the approximate age of our human cohort. And that the young mean age was about 7, and this equated to about 23 human years. And the old were around 21, which equaled about 64. So approximately the age of our human cohort. And we had n equals 10 for each of our groups. And first off, all the microRNAs that we tested by real-time PCR in monkey serum in human serum were also present in monkey serum. And we saw similar patterns as far as 181A was by far the most highly expressed in both monkey and in human. And we found also significant trends in the fact that 181A5P and 1248 were significantly decreased in older monkey serum compared to younger monkey serum. And we also saw a close to significance trend in 181A5P. So we saw very consistent results in both non-human primates, so the Rhesus monkeys, and in human serum. And what's interesting about 181A5P is that we found previously that this was decreased with age in PBMCs in humans. Other investigators here at NIA-IRP found that it's decreased in skeletal muscle in monkeys, and other laboratories have also found it's decreased in the mouse brain. And 181A5P has previously been shown to target inflammatory cytokines. And so we looked at, screened a number of different pro-inflammatory and anti-inflammatory cytokines in the serum from our young and old cohort. And with decreased levels of 181A5P, we saw a positive correlation with anti-inflammatory cytokines TGF-beta and IL-10, and a significant negative correlation with the pro-inflammatory cytokines IL-6 and TNF-alpha. And this would be serum protein levels of these different cytokines. And at this time period, there was very little that was known about miR-1248 and its targets. So to get a better idea of the targets and pathways that it might be regulating, we overexpressed miR-1248 in cells. And we performed a genome-wide microarray to get an idea of the genes that may be regulated by miR-1248. And we focused here, as I'm showing here, on the genes that were negatively regulated, so they were downregulated. And this is because, in general, most microRNAs negatively regulate their target genes. And we found, interestingly, that the age-associated cytokines IL-8 and IL-6 were downregulated in cells overexpressing miR-1248. And indeed, if we do real-time RT-PCR validation for the targets that we found that were regulated by miR-1248, indeed, IL-6 and IL-8 are downregulated with miR-1248 overexpression. And we also did pathway analysis to get an idea about what pathways may be regulated by this microRNA. And here again, I'm focusing on the pathways that were downregulated when miR-1248 was overexpressed. And we found that miR-1248 modulates a number of different inflammatory pathways, including NF-kappa B, various cytokines, TNF receptor 2 pathways, and also increased DNA repair pathways. And to further define whether miR-1248 directly targets IL-6 and IL-8 mRNAs, we biotin labeled miR-1248, did precipitations from cells, and then isolated the RNA and did real-time RT-PCR. We found that IL-6 and IL-8 and also CDK2 were enriched in the biotin pull-downs with miR-1248, indicating that this microRNA can bind specifically to these mRNAs. So what we think is that microRNAs can be important modulators of inflammation with age, and that we found that there were decreased serum microRNA levels with age. MicroRNAs can be important modulators of inflammation, and that changes in microRNA levels with age may explain chronic inflammation observed in the elderly. For example, here's it shown in just cartoon format. So if you decrease microRNA levels in aged individuals, this may cause an increase in inflammatory cytokines. So here we initially focused on microRNA levels in serum. So we wanted to expand this to look at all extracellular RNA in human body fluids. And again, we focused on serum since this is readily bioavailable in our studies. And we wanted to, with the goal of these studies, to profile all extracellular RNA, mRNA, long non-coding RNA, and other small RNAs, such as microRNAs and other RNAs. And we used a slightly different approach in this study because we wanted to initially profile serum extracellular RNA from a larger age span cohort. And so we chose individuals that were from the Handle study and also the Baltimore longitudinal study of aging, which is also an NIA-IRP funded study that's been going on for decades, actually. And both of these studies are here at NIA-IRP, and we chose individuals that came to the clinic approximately the same time frame, and also both of these studies, the core lab processes the samples from both of these studies. And here we actually chose African-American females that were 30 for our young cohort, N equals 13, and older African-American females that were 81 years of age, and there was N equals 10. And so our hypothesis was that age-related changes in extracellular RNAs are important for normal aging and may be important for age-related degenerative processes and diseases. So here's our sequencing workflow, where we had serum from Handles and VLSA participants. And we isolated total RNA using a double trizol LS chloroform extraction from 200 microliters of serum. And we fragmented the RNA into approximately 200 base pairs in length. So this would enable us to sequence in one sequencing reaction RNAs, such as microRNAs, and longer RNAs, such as mRNAs and long non-coding RNAs, again, in one sequencing reaction. And we did library preparation using Ion Total RNA Kit version 2, did template preparation using OneTouch 2, and we sequenced using the Ion Proton Sequencer. And here, just some description of the cohort. Again, these are all African American females. From the Handle study, they were 30 years of age, approximately on average, and from the BLSA study, they were 81. So the younger cohort had 7.95 million sequence reads on average, and from the older group, there were 8.90 million sequence read on average. And we excluded any individual that had less than 1 million sequence reads. So here's our analysis workflow, where we have FASTQ files from the sequencer. We filtered them, and then initially we aligned to human genome version 19 using star aligner from ENCODE with parameters optimized for long transcripts. We assembled into transcripts using cufflinks, and this result was a known and novel transcript. We also took these filtered FASTQ files and aligned them to the human genome version 19 using star aligner optimized for short transcripts. We aligned the microRNAs using MIRD2. We looked at microRNA structure using RNA fold, and the result was known and novel microRNAs for structure. So if we look at the numbers, if we look at linear and microRNA transcripts together, in the young individuals, there were 826 that were unique to young, 1,135 that were unique to old, and 525 that were shared amongst young and old groups. And I should mention that these were transcripts that were detected in 90% of the samples. So here's a donut graph where we're looking at the distribution of extracellular RNA biotypes by individual. So this is a very complicated graph, but I'll try to walk you through it. So each of the individual little donut rings corresponds to the extracellular RNA biotypes from one individual. So in the young, there's 13 rings, and in the old, there's 10 rings. And I've highlighted some of the most predominant or interesting biotypes. For example, here's ribosomal RNA, which is the most predominant. There's also protein coding, which is this orange color here and here. There's snow RNAs, which is this light peach color right here, mitochondrial ribosomal RNA, and miscellaneous RNA. And this is just to give you kind of a visual of how the extracellular RNA biotypes differ by individuals. And also I should mention that microRNAs were not included in these graphs because of the different sequencing analysis workflows. And then also that we didn't combine any of the extracellular RNA biotypes. These were all the ones that were from ensembles. We didn't combine any. So again, here's a view of how they look by individuals. So how do they look if you average them and look at age-dependent differences? So here in green, I know this might be hard to read over here, but what this graph is just basically highlighting is that if we looked at the different biotypes by age, so if we combined and looked at young versus old, we do see differences with age. There's some that don't change, like for example, the protein coding looks pretty much similar between young and old. So this just gives you a snapshot view of the averages between young and old in the distribution. But we were able to see significant changes in transcript biotype with age. So here we see a significant increase in mitochondrial tRNAs, mitochondrial ribosomal RNAs, and also unprocessed pseudogenes. And we see a close to significant trend in an increase in miscellaneous RNA and also a decrease in snow RNA, which was that peach color a couple slides back if you remember. So we are able to see differences in transcript biotype with age. So we looked at specific selected transcripts that in our sequencing look like they may have age-dependent effects, and we did this in an expanded cohort by doing real-time RT-PCR validation. So in this cohort, we have 39 young African-American females and 20 old African-American females. And we see significant changes in a number of the different transcripts, including HBB, which is an mRNA. We have a SNO RNA that we see a significant decrease, and we see an increase in a specific pseudogene, and a small nuclear pseudogene, actually, we see decrease. And then we've also begun to look at the microRNAs that were in this sequencing, and we're going to do more bioinformatic and computational biology, computational analysis similar to the one we did with linear transcripts. This project is unpublished and ongoing, so this is still a work in progress. But we did do real-time PCR validation and found that there were several microRNAs that were significantly changed, including 191.5p, 30E5p, 3607, 320, 425.5p, 145.5p, and 6087. So we are able to see age-associated changes in extracellular RNA. So RNA in the circulation could be bound to cholesterol, could be bound to RNA binding proteins, or encapsulated in extracellular vesicles. So here I'll use the term extracellular vesicles to encompass exosomes, microvesicles, and apoptotic bodies, as the isolation techniques we're using does not differentiate between these three different subtypes. We became interested in EVs because their potential use as biomarkers, and that differences in EVs with various diseases were found, including cancer, cardiovascular disease, and neurodegenerative diseases, and then other conditions such as pregnancy, exercise, and infections. However, most studies thus far had examined EVs secreted from cells in tissue culture, or would focus on a specific disease versus matched control. So we wanted to characterize EVs with human age, to ask the question, do EVs change with age, across the lifespan, over time, concentration, size, or content, including protein and RNA? So to ask these questions, we chose 74 individuals from the Handle study in 3 different age brackets. They were young, 30 to 35, and this was 30 participants, middle-aged, 40 to 55, and this was 30 participants, or old, 55 to 64, and this was 14 participants. And in these categories, all the participants were race and sex matched. And we chose individuals that we obtained plasma from 2 different time points, and these time points were approximately 5 years apart. So this would allow us to do 2 separate cross-sectional analyses and also longitudinal analyses. So we had 148 samples that we isolated extracellular vesicles from, and again, this was from 74 individuals, and we measured concentration and size using nanoparticle tracking analysis. We spent a lot of time and careful consideration into what type of isolation techniques that we wanted to use to isolate extracellular vesicles. So here we have pooled samples of plasma that we isolated either using ultracentrifugation, exoquick, or size exclusion chromatography. And here above each of the graph is the coefficient of variance. So we found that the lowest coefficient of variance in the isolation technique was by exoquick. And also exoquick will allow us to isolate EVs in a more high throughput manner, and would also allow us to look at downstream approaches, especially looking at protein and RNA. So initially using this approach, we characterized our EVs according to the ICES guidelines, and that we first immunoblotted the extracellular vesicles for known EV markers, flotilin-1, TSG101, and CD9. So all 3 of these EV markers were enriched in our EV samples from young or old individuals compared to EV-free. We performed electron microscopy and could found that there were clear membrane vesicles in the size range of EVs between 50 and 250 nanometers. We also performed nanoparticle tracking analysis for both of our cohorts. So this would be visit 1 and visit 2. And our size distribution showed a very similar size distribution with a peak about 150 nanometers, which is typical of EVs that are isolated from plasma. So initially we looked at whether there are differences in EV size with human age, but we were not able to find any significant changes with EV size with age, and we tried really hard. We looked at the average size, we binned them by different sizes, and we also grouped them, but we didn't really see any significant trends. Like we might have seen a difference in one bin, but it wasn't a consistent change. So we didn't see any differences in size. But what about concentration? So here is the cross-sectional analysis. So at visit 1 and visit 2. And again, we isolated EVs from plasma from individuals across the lifespan. We found that in both cross-sectional analyses, we see a decrease in EV concentration with age. And this decrease is observed using linear mixed model regression to account for matching. And so we found that there was decreased EV concentration with age. So again, we obtained plasma from individuals from 2 different time points. So this would allow us to look at longitudinal changes, so changes over time. So this is a spaghetti plot where the point here is the initial concentration at time point 1, and the point here is the concentration at time point 2, and the length of the line is the length of the time. So what we found is that over time, there was a decrease in EV concentration. And what's unique about this type of approach that we had was that we had obtained plasma EV samples from one individual over 2 different time periods, and very little is known about how an individual's EV profile changes. And so we decided to, this gave us a unique opportunity to look at how EV profile changes over this time period. And so we initially looked at concentration, and what we found was that EV concentration is highly correlated between visit 1 and visit 2. So this suggests that an individual has a specific EV concentration profile that's specific to each person. So when we're measuring EV concentration, this is at a snapshot in time. So decreased EV concentration with age could be due to a decreased release from cells or an increased internalization from cells. So we decided to focus on this latter idea first. So we developed a fact-based assay to test this. And we had isolated EVs from the different individuals across the lifespan, and these were all individuals from the same cohort that I talked about previously. We labeled them with fluorescent dye, and then we isolated freshly isolated PBMCs from a single individual. And these, the circulating cells would be B cells, monocytes, and T cells, because we hypothesized that circulating EVs and plasma would be interacting mostly with these cell types. And we incubated the EVs from different individuals with PBMCs, and then we did FACS for PBMCs that were positive for EVs. We use CD3 as a marker for T cells, but we also use in some cases CD4 and CD8. We use CD19 for B cells and CD11B and CD14 for monocytes. So at the time when we were doing these experiments, there was very little known about what cells would be internalizing EVs in the circulation. So we had to do a number of different controls that I'm not going to talk to you about today, but I'd be happy to answer any questions about them later, or you can certainly email me. But what we found was that monocytes were the predominant cell types that internalize EVs, as shown here on the FACS. Sorry about that. Or it's quantified over here, followed by B cells, but that the internalization of EVs by T cells was negligible, which is actually somewhat surprising since T cells make up the majority of the cell types in the circulation. And here again is the quantification of these results in a number of different PBMC donors. So to test whether this mechanism of internalization was passive or active, we treated monocytes with latrunculin A, which inhibits endocytosis, and found that in incubation with latrunculin A totally abolished monocyte internalization of extracellular vesicles, suggesting that the EV internalization by monocytes is in part regulated by endocytosis. So the question we really wanted to answer is the decreased EV concentration with age due to increased internalization by circulating cells? So we have the EVs that we isolated from the individuals across the lifespan. We incubated incubated them with the PBMCs, and then I did our FACS-based assay to look for internalization. And here I'm showing the B cells internalization, where B cell internalization increased as the age increased. And this was consistent among the 4 different PBMC donors that we tested. We didn't see any significant differences in monocyte internalization with the age of the EV donor. So just to summarize, it shows that there's increased internalization of EVs from older individuals in B cells, and that this may account for the decreased circulating EV concentration that we observe with age. So we wanted to look at, does EV internalization activate B cells and monocytes? So to do this, we looked at the cell surface expression of CD25 on B cells and MHC2 on monocytes. And we had similar results looking at CD80, a co-stimulatory molecule as well. So cells that had internalized EVs were more likely to have CD25 expression for B cells and also MHC2 for monocytes. So EVs are able to activate B cells and monocytes. And what about, does this change with the EV donor age? So here again is a plot showing the EV donor age, and we looked at CD25 mean fluorescent intensity. So how bright is CD25 on the cell surface? So the age of the EV donor did not seem to affect CD25 expression on B cells, but it did affect MHC2 expression on the cell surface, and that EVs from older individuals increased MHC2 on the cell surface. So we hypothesized that changes that different EV proteins may account for the differences in internalization. So we wanted to screen a number of different proteins in the extracellular vesicles. So we did this using 2 different approaches. One was an EV array in collaboration with Melina Jorgensen's group at Aalborg University, and this looked at 37 different proteins. And we also lysed the EVs and looked in ELISA-based assays. And here I'm just going to show you the significant results that we found, and that we wanted to show, to look at protein changes that were consistent. So they were consistent amongst our longitudinal experiments, our cross-sectional analysis, and our cross-sectional visit 2 analysis. So here I'm highlighting 5 different proteins that were significantly changed in all 3 different parameters. And these were in and also in the same direction. So in all 3, we found an increase in CD151, which is a tetraspanin that's important for many cellular processes that are important for tumorigenesis. We also found an increase in mucin 16. Mucin 16 is actually also known as CA125, and it's a circulating marker of, a well-known circulating marker of ovarian cancer and other types of cancers. And I should mention that it wasn't observed, that we didn't measure this in all individuals. There's only a limited number of individuals that had expressed mucin 16 on their extracellular vesicles. And we also found that there was decreased apoptosis markers, cleaved PARP and cleaved caspase-3 and P53 with human age. And again, we use this longitudinal approach, and this allows us to compare protein profiles from year 1 to year 5. And what we found was that there's a significant Pearson correlation comparing visit 1 and visit 2 for 39 out of the 46 different proteins. So here's just a little graph. I didn't show you all the information, but it's published, it's shown in this report here if you want to look back. So this suggests that each individual has a specific EV protein profile that doesn't vary that greatly over time. So just to summarize these results, we found that extracellular vesicles change in concentration and protein content with human age. And I didn't have time to tell you, but we also found that demographic factors or lifestyle factors such as BMI and smoking may also affect EV concentration. And then if we look at our longitudinal analysis comparing year 1 to year 5, this analysis showed that each individual may have a specific EV protein profile and concentration profile that may be unique to this individual. And so here, just to summarize the rest of my talk, what I've showed you is that microRNAs decrease with human age in peripheral blood mononuclear cells, possibly through downregulation of the dicer, the microRNA processing protein. There were specific serum microRNAs that are decreased with age in humans and in monkeys. And then unpublished work that's still ongoing, we identified that there may be extracellular RNA transcript biotypes that change with age, and specific transcripts that may also change with age. And that we also found that there was decreased EV concentration with human age, and this may be due to an increase in B-cell internalization of older EVs. So I'd like to thank a number of individuals. So I am a staff scientist in Dr. Michelle Evans' lab. She's the Deputy Scientific Director here at NIA-IRP. We have a number of different National Institute on Aging Intramural Research Program collaborators within our department, such as Alan Zonderman and Nicole Noren, and then also in other departments, such as the Laboratory of Neuroscience, Rhesus Atan and Mark Madsen. We developed the FACS-based assay with Monica Bodogai, and that all the sequencing work was done in collaboration with a very talented core facility in the Laboratory of Genetics and Genomics. And the BLSA samples are from the Translational Gerontology branch. And we also have other collaborators outside of NIA-IRP, including Melina Jorgensen from Aalborg University and Ken Whitwer from Johns Hopkins University. And also you can feel free to ask questions after the webinar. Here is my email address, so you can feel free to email me. And I also will be at the November Extracellular RNA Consortium workshop. So you can feel free to grab me there too. Thank you.
Great, thank you, Nikki. Very interesting. So, Nikki, regarding the age, you said that these decrease with age. And what's known about their potential involvement with various diseases which, accumulate with age, whether it's arthritis and other type of diseases? Any correlation or what's known with their potential impact?
Are you referring to the extracellular vesicles or the microRNAs or the extracellular RNA?
Well, I guess both. I think in EVs and the microRNAs in general.
MicroRNAs, yeah. So, it's still, as you know, in the Extracellular RNA Consortium, it still is a very wide open field on the microRNAs in serum. So we actually wrote a review that was published this year in Wired's RNA, where we looked at a bunch of the different, RNAs, microRNAs that change with age in the studies that have been performed. I mean, the hard part with this field is that there's not a lot of reproducibility amongst the studies that have been looked at, or some things have been done in mice, some things in humans, and that doesn't always relate. But there are specific microRNAs that show a pattern to be decreased with age, and that may also be important for various other diseases. I mean, I think of a large global analysis looking at the microRNAs that are decreased or change with age in relation to some of the other age-related diseases has not always been done, in a great detail. But there definitely are similarities in some of the microRNAs. For example, miR-151A5P, which is decreased in our study, correlates with the survival factor of one or two specific cancers. But at that time when we were doing the study, it really was very little known about it. And it's known that miR-181A, is decreased in a number of studies with age and does affect inflammation. And that one does keep popping up as something similar, reproducibility.
And this study, going into communities, that's ongoing?
Yes, that's ongoing. So it's a longitudinal study. So it was started in 2004, and it's ongoing. We're getting ready to start wave 5. So each of the individuals, it's a longitudinal study. So we visit each individual approximately 5 years apart, depending on where we go in the neighborhoods and everything. So we're on the wave 5 of data collection. And data collection from each. It depends on each wave on what we collect, but we always collect plasma and serum and PBMCs. And then some of the other biofluids we collect depending on the different waves.
Okay, thank you.
Nikki, this is Julie Sogsted calling, that was a wonderful presentation.
Okay.
I was a little surprised to see the mitochondrial RNA, tRNA, and small RNA transcripts increase with age. Can you comment on what you think might be happening in that situation? That was interesting, I thought.
Well, I think yes, we don't really know. It's still very early on, but there is some data that mitochondrial and portions of mitochondria are actually present in extracellular vesicles. Why this may change with age, we don't know. It could be that there's damaged mitochondria, so maybe and that's one thing that does happen, we know, with age, and so maybe the cells are trying to, I think it's still debatable now in the circulation. If you find something in the circulation, is it because of the cells are damaged? Or, like, for example, we see decreased apoptosis proteins in the extracellular vesicles with age. Is this because there's decreased apoptosis, or is it the decrease of the actual getting rid of the garbage, for example? We don't really know, and I think that's still debatable. But there is a large, and it's also becoming a lot more popular right now about mitochondrial effects with aging. So it may, so that may be something. And these are also some of the RNAs are non-coding, and so we don't. There's a lot of differences with that too. So I think it's something that we're definitely interested in pursuing.
Yeah, I thought that was very interesting. So, but very nice presentation. Thank you.
Thank you.
Other questions for Nikki? Nikki, Matt Roth had one other. You talked about some of the methods you've used. Have you explored, or would there be utility for vesicle flow cytometry approaches to help you sort some of these populations?
I think definitely, because as you see, when we tried to look at some of we're using some other different approaches to look at proteins, but looking at proteins is very laborsome, to look at the different proteins. So I definitely think that vesicle, using flow cytometry may definitely be something that's worth looking at. As long as you have obviously the technology and the instrumentation to look at the small vesicles, I think that would be very powerful. And it also needs to be sensitive enough to detect the small amounts of protein.
All right, thank you. Okay, any last questions for Nikki?
If not, feel free to email me, or I'll see you at the Consortium event as well.
Yeah, great. No, thank you again, Nikki, and we will look forward to seeing you in November. Thank you very much.
Thanks.